Good morning, biology, and welcome back to Chapter 17, which is on the classification of organisms. Today we're going to talk about different ways that we have kind of went about classifying all the living organisms on the planet and kind of the current systems that we use today. All right, so let's jump right into it. All right, the first thing here is biodiversity. We have a very diverse planet with lots of living creatures on it. Currently, we have named over 1.3 million species, and that's only the ones that we have found, discovered, and like really well identified. We estimate that there's around 8 million alive today and we have yet to discover that other 6.7 or excuse me 6.7 million species. So biodiversity itself is just the variety of organisms that we see. It's at all levels, from population to individual species, all the way up to ecosystems. And how we classify 1.3 million things is a challenge. It becomes a kind of a conversation because it's a lot of things to break down and kind of classify. But to do this, we kind of created this science called taxonomy, which is the science of describing, naming, classifying organisms. And on a taxonomic group here, we have taxons, or any group here that is a kind of the original, is the end group. We call these the leaves of the taxonomic tree. The branches are like these kind of breaking points here. Nodes are where they connected and then broke apart. So those are kind of some basic terminology there. Historically, for taxonomy, we had Aristotle. And Aristotle took everything and he classified down to two things. Either it was a plant or an animal. He then grouped them, whether they lived on land, whether they lived on water, whether they lived on the air, whether they were small, medium, and large for plants. And this worked really, really well when there was very few organisms. The ancient Greeks and Aristotle could only see organisms native to the Mediterranean and the surrounding area. They didn't have millions of species to kind of catalog. But we found out that these names aren't super, super useful and that common names aren't really useful. And we'll give an example right here. What is the name for this animal? Well, if you're watching this in North America, or excuse me, like the Pacific Northwest, you're probably gonna say this is a mountain lion. If you were down south, say it was a puma. Some people would say it was a cougar. Some people might say it's a panther. Science has kind of made this a little bit easier. We refer to this animal in the scientific community as puma concolor. Sorry, that was very fast, but that was puma concolor. So, Linnaean Classification, it was created by this guy named Carolus Linnaeus. This is 1707 to 1778. He created a grouping system for organisms, started with very, very general categories, and narrows an organism down to smaller and smaller specific layers until we finally get to the species layer. And it uses 8 layers, and we're going to talk about that for puma concolor. So, for our little guy here, he is in domain Eukarya, kingdom Animalia, phylum Chordata, meaning he has a backbone, or develops that way. Class 4, he's in Mammalia, which means that he has fur and produces milk. Order Carnivora, meaning that he only eats meat. Family Felidae. Genus Puma. And species Puma concolor. When we write the species name, we also write the genus name with it. So, every species has 2 unique names, 2 unique parts of it. It has the genus name and then the species identifier. And we call this system binomial nomenclature here. So Homo sapiens for us is the smart apes, and we have 2 names. Homo is our genus name, and then sapiens is our species identifier. So when writing names, our genus is always going to be capitalized, and then the species name will be lowercase. So genus, species. And then I'm really bad at using this tool and drawing in italices, but anytime that you draw these or write these or especially type these, you should have the italics for the genus species. So the names are usually Latin, and that's because Latin is a dead language. It doesn't change, so there's not like we're not getting new words inside of the Latin language like we do inside of the English language. So that's why we use Latin. All right, next up, we talk about systematics. This is a grouping or a system or some kind of classification method to organize relationships in animals. This is a whole science behind this. The similarities can be a lot of different things. It can be things like taxonomy. We're talking about the description of that organism. It can be classification. It can be nomenclature, like the naming of them. And then morphology, which is these similarities like visual, like the terminology of their parts. So it's breaking up this organism and classifying it based on what it has. So you can use data that's embryonic, you can use chromosomes, you can use proteins, you can use DNA. All of that is a fair game for systematics to make these kind of connections between organisms. All right, phylogenetics. This is the analysis of the evolutionary or ancestral relationship among taxa. So we look at this one, we're very interested in what came from what and kind of what the direction of life is going. So we'll use lots of different pieces of information for phylogenetic trees. We can use visual similarities from fossils. That's kind of how we started. We're moving towards things, looking at things like embryo development here. These are all vertebrates here. And we can see how they all kind of develop the same at the beginning and then differentiate later. And we can look at gene expression, how they create and have genes. We can look at like myoglobin in humans and mammals, how we have this muscular blood carrying thing that's red versus other organisms. When we get enough of these broken apart, we can create what's called a phylogenetic diagram or a phylogenetic tree, where we break things up based on certain characteristics, and we have this now tree here. It looks like a family tree, but different branches or different species instead of different like family members. If you're looking for humans, we're out here. All right, so to form these, we're going to look at evidence of shared history. So we commonly use fossils to study evolutionary similarities, but there are some gaps in the fossil record, especially for soft or small-bodied organisms, things like bacteria, things like worms, things like fungus. All of these we might consider worms or have worms in the name, like roundworms, uh, worm eels, like flatworms. This is like in the same group as earthworms here. So there's a lot of worm organisms here. These guys are very soft-bodied. They don't leave very good fossils, so we don't have a lot of record of how they have changed over time. We'll also use things like homologous and analogous structures to group organisms, and we'll do a real quick review on those. Remember that homologous are from the same common ancestor, things like the forearm bones of human, cat, horse, bat, and dolphin, that have different features now in the kind of final form. This came from the same ancestor, but now it's used much differently in all of its relatives. And then analogous features here are ones that came from different ancestors, but have or look the same and do the same purpose. A group that we're really interested in are the amniotes. This is a group like us that have a fluid-filled sac called an amnion, which is what we put our young in. And these are things like reptiles, mammals, and birds. And the other vertebrates lack an amnion, so things like fish and amphibians don't have this. It's important to talk about cladistics in this kind of discussion. In 1966, German biologist Willy Hennig here, he developed the science of cladistics, which is a system of phylogenetic analysis, so using those relationships, using only shared and derived characteristics. And what does that mean? Well, shared versus derived characteristics here. Shared characteristics are a feature that all members of a group have in common. So example, all mammals have hair, all birds have feathers. But we can also look at derived characteristics. This is features that evolved only within the group under consideration. So even though all birds have feathers, so do ancestral dinosaurs. So now that's a derived characteristic. We're looking at the relatives of that organism and seeing how that trait has derived itself in that line. All right, clade. In cladistics, a clade is any organism, let's say it's this chimp here, and their ancestor. So it'd be this guy down here, their ancestor. Cladigrams don't use names like phylum or class. They are looking at more specifically traits. So all organisms, once again, are grouped to their ancestral species, and then we can use a cladogram here to look at derived versus shared characteristics. Everything past this line here has jaws. So the salamander, the perch, the lizard, the pigeon, the chimp. Anything past this line here has claws or nails. So this is uh, kind of like the trees that I had you do already. All right, molecular cladistics. By counting the shared or derived amino acids and proteins, we can construct a tree uh, based on molecular cladistics. So what this would mean is that organisms coming from more recent common ancestors should have closer DNA, and the closer the DNA of two organisms are, the closer they should be on a cladogram. So we'll look at proteins, kind of like this is here, and you can see that the chicken has a little bit different protein here, it has a different protein here, it has a different protein here. This means that the chicken is probably not as closely related to any other group on here. But if you want to look at ones that it is close to, probably closer to things like the mouse and the rat that it has similar genetics to. So, really, really cool branch of science. A lot of work being done here. Probably the most work being done here. So, very exciting stuff. All right, the last part of this is going to be on modern classification. We're going to look at remember that the original tree of life that we had for Linnaeus had two main groups: eukaryotes, which have a nucleus and membrane-bound organelles, and prokaryotes, which are these bacteria and archaea here, that have no nucleus or membrane-bound organelles. So, until the mid-70s, we had everything broken into 6 kingdoms. We realized, hey, this is not a super accurate way to classify organisms. So we had classification systems such as like complex and simple, but we discovered that bacteria, all of these are bacteria here, and different species of them are very diverse and actually very complex in their own way. The things that they're able to do, I mean, they're able to break down molecules and substances that we're unable to. They're able to break down oil and oil spills and use that as fuel. So we kind of got over our ego a little bit and decided to call these not simple anymore. They have their own complexities. What we did do is we broke into 3 domains of life, and there's 3 kind of important points here for why we did this. The first thing is all living creatures inherited their rRNA, their ribosome genes from an ancient ancestor. We call this ancestor the last universal common ancestor, or some people will abbreviate it to LUCA. So now we have broken it, and all living things are related by ancestry to 3 domains. And they are bacteria, archaea, and eukarya. And the third thing is, and you guys have already learned this, but this is new for anyone that learned since the 70s, is that archaea is not the same as bacteria. Although they look similar, they're single-celled, they have no membrane-bound organelles, they have no nucleus. This group came from a different ancestor than bacteria. So this is our tree here. As you can see, somewhere right here, an ancestral last common universal ancestor was here. It broke off to become the bacteria, and it broke off to become the archaeans. And then we broke off from the archaeans to become the eukarya out here. We're going to real quick go through the domains and the kingdoms, give you guys a little bit of insight into them. The first is domain bacteria, and like I said, there are lots of different types of bacteria. They are not simple, quote unquote, they are a very complex group. They are made of small, single-celled, prokaryotic organisms, meaning no nucleus and no membrane-bound organelles. They usually have a cell wall, and they will produce by binary fission. So they lack complex organelles, and they usually have one circular chromosome. Archaea, these are also prokaryotic. They don't have the cell walls usually as often, but they do have cell membranes that are very unique to them. Most of them are autotrophs, making their own food by chemosynthesis, and they can live in very, very extreme environments. And they're thought to be among early Earth's inhabitants. They lived in deep sea vents, they lived in hot springs, they live in salty lakes now, they live in animal intestines. There are no parasites of these. We don't get sick from archaeans, so we kind of like them. But we believe that the symbiosis bacteria swallowed one of these guys and created domain Eukarya. This is the group that you're probably most familiar with. They have very, very large cells. They have nuclei inside of these cells. They have complex cellular organelles. Inside of this group, we have plants, fungus, animals, various different single-celled organisms. And we're going to go through each of those. So we have 6 kingdoms, which was the original kind of way that we split up life. We now break it into the different domains. So the domains are higher. So it goes domain. And then underneath of that, kingdom. And you'll get practice with this because I want you to be able to be proficient at scientific names. Alright, so kingdom Eubacteria here. This is lying underneath domain bacteria. Eubacteria means true bacteria. This means true. We no longer consider archaeans in this group. They get their own. And kingdom Archaea is nestled under Archaea. It means ancient bacteria. But now lots of scientists only use Archaea. I'm among them. We don't really use Archaebacteria anymore. Archaebacteria and bacteria both only have one kingdom because their domain has all of the things that are underneath of it. But now, kingdom Protista is inside of the eukaryotes here. Eukaryota. And we call these groups the protists. And these are eukaryotes that aren't animals, plants, fungus, live inside this group. It's not a great kind of way to break up organisms because they're based on what they're not, based on what they are. But we have lots of groupings inside of Protista that make more sense. Things like the algae, things like the flagellates, and things like that are more straightforward. So some groups here, amoebas, paramecia, euglena, if you remember from the pond water lab, you've probably seen some euglena in there. We call them simple once again. They're very much smaller than big eukaryotes. I wouldn't say more simple. All right, the next is kingdom fungi. These are heterotrophic eukaryotes, meaning they can be either unicellular or multicellular. The important thing about them is they absorb their nutrients. That's how they get their food. There's no photosynthesis. They don't eat things, they absorb them. Things inside of this group are things like mushrooms and rusts and smuts and mildews and molds. All of these up here are examples of fungi, even though this is like the most common way we think of it. Most fungus lives like a mold or a yeast. All right, kingdom Plantae. These are also multicellular eukaryotes. They're autotrophic or photosynthetic. Things like, there are some examples that are not, though. Things like mistletoe is an example of a parasite instead. Plants develop from embryos. These are what's inside of seeds, and they include things like the mosses, ferns, conifers, and flowering plants. And then the last group is kingdom Animalia. These are multicellular eukaryotes. They're heterotrophs, meaning they have to eat other food. They can't make their own. They develop from embryos, and they have body symmetry and can move, is important things about animals. So, these are a smattering of the animals that we see. Very, very exciting, very, very cool. And this is the end of chapter 17. So, if you have any questions, let me know. I hope you learned a lot about the classification systems, and we'll see you in the next one.